Hello Copperheads, my name is Cupri, welcome back! If you don't know what Wilson's disease is, or if you need a refresher, you can go watch my introduction video by clicking somewhere around here on the screen. I'm sorry, I'm new to YouTube. This is probably the longest and most complex video I'm ever gonna make for you. But I believe you can learn this because you can learn anything you want. And if you need to rewatch something or take a break and come back later, please do it. That is the beauty of using videos. You can learn at your own pace. As you know, Wilson's disease is a rare genetic condition of copper metabolism. Specifically, it's an autosomal monogenic recessive condition connected to gene ATP7B. Next week, I will tell you more about this gene, how it works, what it does, and why it is connected to this condition. But today we're going to focus on the genetic inheritance of Wilson's disease as an autosomal monogenic recessive trait. If you have no idea what I just said, don't worry, because in a few minutes you really will. Important note before we get into it. Human genetics are actually a lot more complicated than I can explain in one video, so today we're only gonna cover what usually happens and not all the possible but very unlikely exceptions. That would be advanced genetics, which I do plan to tell you about one day, but a bit further into the future. So let's focus on the now. This video has 3 parts. On the first part, I teach you basic concepts about genetics. In the second part, we look at a simple example of an autosomal monogenic recessive condition. And in the third part, we learn about the specific case of Wilson's disease. Okay, ready, set, go! Part 1. Basic genetics. Imagine there is a furniture encyclopedia. It contains instructions to make any piece of furniture you could ever need. No pictures, just words. Some instructions are simple, like how to hammer a nail, and some are complicated, like how to assemble a bed. Some instructions are general and can be applied to anything, like how to cut some wood, and some are specific, like how to install the hinges in that one cabinet. Every bit of knowledge you need, it's all in there. So obviously that's a lot of information, which is why this encyclopedia is split into a number of books, specifically 23. And inside each book there are hundreds or even thousands of sections, and they are all written out using the letters of the alphabet. Now, you actually have 2 copies of the encyclopedia, one from each parent. The copies have been passed down for generations since the very beginning of furniture making. But keep in mind that when a child is born, the parents still need to keep their own copies of the encyclopedia to keep making their own furniture. And in the world of my little analogy, there are no photocopy machines, so they need to do it by hand. Can you imagine how many misspellings or simply different spellings accumulate when people are copying by hand over and over again a text that is billions of letters long? That means that the copy of the encyclopedia you got from one parent is gonna be somewhat different to the copy you got from the other parent. Alright, so this is what your DNA is. It's the huge instruction manual that makes you. Now pay attention because I'm gonna tell you some important genetic terms. Your 2 copies of the encyclopedia together would be your genome. Each book would be a chromosome. Each section would be a genetic sequence. There are different kinds of genetic sequences, but today we will only talk about genes. And the letters are called bases. There are 4 bases in human DNA. They have longer names, but we can simply call them A, C, G, and T. The fact that there are differences between all the copies of the encyclopedia out there would be called genetic variation, and each individual misspelling or different spelling would be called a genetic mutation. Let's review the important terminology: genome, chromosome, gene, base, genetic variation, and genetic mutation. Got it? Awesome! Part 2. Basic autosomal monogenic recessive diseases. Before you existed, there was an egg and a sperm. Each of them contained 23 chromosomes. Chromosomes 1 to 22 are the non-sexual chromosomes, also called autosomes. And the 23rd chromosome is called either X or Y and is the sexual chromosome, also called allosome. The egg and the sperm fuse together to make a full cell with 23 pairs of chromosomes, so 46 in total. This initial cell basically cloned itself many times until eventually there was you. You might be wondering, if all the cells in my body are basically clones of each other, how come the cells on my skin look and act totally different from the cells on my eyes? Well, that would be because even though they all have the same genome, they don't express the same genes because they have different functions. To go back to the encyclopedia analogy, you don't build every piece of furniture in the book every time you open it. You need a table, you make a table. You need a couch, you make a couch, and so on. Let's talk about autosomal monogenic recessive diseases. Autosomal means that the gene or genes responsible for the disease are on the autosomes, the non-sexual chromosomes. Monogenic means that there's only one gene responsible for the disease. And recessive means that you need two pathogenic, bad copies of the gene in order to actually develop the disease. So how does it actually work? Let's look at a simple example. Let's take disease A. It's not Wilson's disease, it's an imaginary autosomal monogenic recessive disease connected to gene A. Gene A has two versions, also called alleles. One allele is big A. We call it the wild type because it's the original, most common, benign, good form of the gene. We write it with a capital letter because it's stronger, in other words, dominant. The other version is small a. We call it the mutant because it's the mutated, less common, pathogenic, bad form of the gene. And we write it with a small letter because it's weaker, in other words, recessive. Everyone has two copies of this gene, one from each parent, so they can have different combinations of big A's and small a's. This is called the genotype, the types of genes they have. They can have two big A's. This is called a homozygous dominant genotype, because homo means same. They can have one big A and one small a. This is called a heterozygous genotype, because hetero means different. Or they can have two small a's. This is called a homozygous recessive genotype. Again, homo means same. The way the genotype actually manifests itself in their body is called the phenotype. If they have two big A's, their phenotype is unaffected because the disease doesn't impact them in any way. If they have one big A and one small a, their phenotype is carrier. Big A wins over little a, so they don't actually develop the disease, but they do carry one small a in their genome and they can pass it on to their children if they have them. Or, finally, if they have two small a's, their phenotype is affected, which means they do develop disease A. Let's use an example of two people who want to have a child. One parent has big A, big A. Their genotype is homozygous dominant. Their phenotype is unaffected. The other parent has small a, small a. Their phenotype is homozygous recessive. Their phenotype is affected. When they have a child, the child gets one big A from the first parent and one small a from the second parent. The child's genotype, big A, small a, is heterozygous. The child's phenotype is carrier. The child doesn't develop the disease because they have one benign, good big A to compensate for the pathogenic, bad small a. But they do have one copy of small a, which they can pass on to their children if they have them. So what happens when two carriers of disease A have children together? To find out, we will draw a Punnett square, which is used in genetics to find out the probability of different genetic outcomes. So we have one carrier over here with one big A and one small a. They will only pass one of the alleles to the child, and this is random, so there's a 50% chance it'll be big A and a 50% chance it'll be the small a. And over here we have another carrier who is just the same. So now we can fill out our Punnett square to see what the different possible outcomes can be. So maybe the child gets one big A from each parent. This child is homozygous dominant and is therefore unaffected by the disease. Or maybe the child gets one big A from one parent and one small a from the other parent. So the child is heterozygous and is therefore a carrier, just like the parents. Or finally, maybe the child gets one small a from each parent. So now this child is homozygous recessive and is therefore affected and develops disease A. Now let's talk about probability. If the big square represents all possible outcomes, each of the equal-sized little squares has a 25% probability or 1 in 4 chances of becoming true. 25% probability of being unaffected, 25 plus 25 equal 50% probability of being a carrier, and 25% probability of being affected. But keep in mind, this doesn't mean that if these two carriers have 4 children, one of them is gonna be unaffected, 2 of them are gonna be carriers, and 1 of them is gonna be affected. No, this means that every individual time they have a child, these are the odds that the child has with respect to inheriting disease A. If 2 carriers have several children, it could be that they're all unaffected, or that they're all affected, or whatever, because it's random every time. Of course, there are other possible genetic scenarios, like what happens when an affected person has children with a carrier. But how about this? Why don't you try to draw the Punnett square and then check your work by using Google or by contacting me? Anyways, the example I made up about disease A is very limited and simplified, because with disease A there's only 2 alleles of the gene, big A, which is the benign dominant wild type, and small a, which is the pathogenic recessive mutant. Furthermore, there are only 3 possible genotypes: 1 homozygous dominant, 2 heterozygous, and 3 homozygous recessive. And there are only 3 corresponding phenotypes: 1 unaffected, 2 carrier, 3 affected. However, real life is not usually like this. Wilson's disease is definitely not like this, but before I explain why, please take a moment, inhale, exhale, pat yourself on the back, because I've already thrown a lot of information at you and you're still here. You are amazing and you should be really proud of yourself. Go Copperheads! Part 3. The basic genetic inheritance of Wilson's disease. Wilson's disease, like most genetic diseases, is much more complicated than that disease A I made up. First of all, there's not just 2 alleles of the Wilson's disease gene, also called ATP7B. There are hundreds. Some of them are benign, good. Some of them are pathogenic, bad. So let's call the benign alleles big W1, big W2, big W3, big W4, etc. We're using big letters because in Wilson's disease, the benign alleles are dominant. And then let's call the pathogenic alleles small w1, small w2, small w3, small w4, etc. And we are using small letters because in Wilson's disease, the pathogenic alleles are recessive. Okay, so let's take a person who is unaffected by Wilson's disease. They don't have the disease and they're not carriers. They have 2 benign alleles of the Wilson's disease gene. The alleles could be big W1, big W1, but they could also be big W1, big W2, big W1, big W3, big W1, big W4, etc. Big W2, big W2, big W2, big W3, big W2, big W4, etc., etc., etc. There are many combinations of dominant alleles that they can have, and in most of these situations, their 2 dominant alleles are non-identical. This means that most people who have the unaffected phenotype don't actually have a homozygous genotype. Think of it this way. Imagine you have 100 pairs of socks of different colors, but they're all loose and mixed up inside of a huge box. If you close your eyes and take 2 random socks, it's very unlikely that they will be the same color. Same goes with someone who is affected by Wilson's disease. They have 2 pathogenic alleles of the Wilson's disease gene, which could be small w1, small w1, but also small w1, small w2, small w1, small w3, small w1, small w4, etc. Most of the time, their 2 recessive alleles are not identical. Therefore, most of the time, people with the affected phenotype don't have a homozygous genotype. There is actually a word to describe people who have 2 non-identical recessive alleles, and that would be compound heterozygote. As for carriers, here's the thing. All carriers are heterozygous because by definition, a dominant allele and a recessive allele are not identical. But not all heterozygous people are carriers, as we just saw that you can have two non-identical dominant alleles or two non-identical recessive alleles. And yet, when most people, including many doctors and scientists, talk about Wilson's disease, they say, if you are affected by the disease, you're homozygous recessive. If you're unaffected, you're homozygous dominant. If you are heterozygous, you're a carrier. So why is almost everyone saying things that are incorrect? And the answer is convention. This way of using the terms comes from the old days of genetics, before we even knew there can be more than one dominant allele and more than one recessive allele of the same gene. Now you might ask, but Cupri, are you nitpicking and just being pedantic about words? And the answer is no. This is actually very relevant because genetic testing is becoming more and more important in diagnosing Wilson's disease. And I will tell you more about this when we learn about the diagnostic process a few weeks in the future. In the meantime, I want to tell you, it's not your fault you were taught the wrong words. So now, what can you do? Well, don't say I'm homozygous dominant. Say I have two dominant alleles. Don't say I'm heterozygous. Say I have one dominant and one recessive allele. Don't say I'm homozygous recessive. Say I have two recessive alleles. This way you get across the important information without assuming that all Wilson's disease patients have two identical recessive alleles of the ATP7 gene. And remember that most sources about Wilson's disease are still gonna use the wrong words. But now you know better. Alright, Copperheads, congratulations! You made it all the way to the end. I'm so proud of you and you should be too. If you want to keep learning, to show me some love or to ask me anything, you can subscribe on YouTube, follow on Instagram, like on Facebook or send me an email. My name is Cupri. See you there, Copperheads!